Hi everyone! In this presentation I will talk about my recent work on intact connectional morphometricity learning using multi-view morphological brain networks with application to autism spectrum disorder. This work has been done at BASIRA lab by myself, Alaa, Bessadok, and supervised by Dr. Islem Rekik. Neurological disorders such as brain dementia affect the morphology of anatomical brain regions to different degrees. As you can see here in this figure, there is an atrophy in the cortex of the demented subject compared to the healthy one. Also, there is an enlargement of the ventricles in Alzheimer's disease patient. Several studies aimed to identify the functional signature of brain disorders. However, a few of them focused on the morphological fingerprint of a specific brain disorder, which can show the association of neuroanatomical changes with function and cognition. So the question here is, how can we discover a disorder-specific morphological signatures? To answer this question, a recent work proposed a statistical metric called brain morphometricity, which investigates the morphological fingerprint of a specific brain disorder, such as Alzheimer's disease. It is defined as the proportion of phenotypic variation that can be explained by brain morphology derived from structural brain MRI scans. In other words, it describes the association between brain morphology and multiple risk factors such as age and gender. However, to identify morphological signature of a specific trait, this work investigates the morphometricity of each brain region independently, which overlooks the relationship in morphology between brain regions. Basically, this cannot reveal information about how morphological changes in region A are influenced by changes in region B on a connectional level. These changes can be described by high-order brain measurements or brain connectivity. More practically, to estimate brain morphometricity, volumetric and thickness measurements from structural MRI are first generated. Then similarity matrix is computed for each of these measurements separately, then averaged to produce the global anatomical similarity matrix. This matrix captures the similarity between brain morphologies of brains drawn from distinct groups, for example, healthy and disordered subjects. However, this matrix is computed using a predefined function such as Gaussian metric that is less robust to outliers, may not handle well multi-view data drawn from multiple sources, and can fail to capture data sample distributions with varying bandwidths. In order to overcome all these limitations, we introduce in this work intact connectional brain morphometricity, ICBM, a new way to estimate the brain morphometricity. First, we use multi-view morphological brain networks, MBNs, that were recently introduced and showed great potential in brain disorder diagnosis to learn a morphological intact space. This can nicely capture the complementarity between different brain networks. Second, our ICBM efficiently learns a similarity matrix by adaptively assigning neighbors for each data sample based on local connectivity. And finally, we compute the intact connectional brain morphometricity that shows how the morphological connectivity fingerprint brain disorders. So, morphological brain network was first introduced in the work of Mahjoub and co-authors in their scientific report paper published in 2018. By learning how to discriminate late mild cognitive impairment (MCI) subjects from Alzheimer's disease subjects, they discovered morphological connectional biomarkers. They basically investigated the morphological connectivities between airways using structural T1 weighted MRI, and they explored the relationship between multiple brain connectivity morphological views, which capture the relationship between brain regions in morphological changes. We use a morphological brain network that can be represented as a connectivity matrix comprising a set of nodes or brain RY, and the connection between two nodes quantifies the dissimilarity in shape between two RYs I and J by computing the absolute difference between RY-based average morphological measurements. For each subject S, we construct a set of MBNs that include different cortical attributes, for example, cortical thickness, circle depth, average curvature, etc. Since each MBN can be defined as a symmetric matrix, we only vectorize the upper triangular part to generate a feature vector FSK for each subject S and each view K. To better represent our data, we concatenate horizontally for a specific subject all feature vectors corresponding to different views. Here I give an overview of the proposed Connectional Brain Morphometricity learning framework, which comprises 3 key steps. The first one is the intact connectomic space learning, the second one is the multi-view morphological similarity learning, and the last one is the ICBM estimation. So let's begin with the first step. To clearly understand the notion of intact connectomic space learning, we provide an example. So, using morphological features derived from different networks independently for dementia diagnosis might fail to capture brain changes in a comprehensive and integral way. So, to address this limitation, one can integrate all features by mapping them into a shared space. Given a set of multiple network views for each subject in a given dataset, we generate a complete intact space X. Specifically, the feature view F1 that represents the view 1 of all subjects in the intact space X is generated via a mapping function W1, which projects each view independently into the intact space. Our problem is formalized as an optimization problem where we define an energy function to be minimized. The first energy term is used to recover the shared space by minimizing the sum of squared distance between the original network's views and the mapped network's views onto the intact space X, using the learned mapping function W. And the second term is used for energy regularization to penalize the intact space. Lambda 1 here is a trade-off parameter. So the second step of the intact morphological similarity learning is the multi-view morphological similarity learning. So we are trying to estimate the similarity between all samples in the dataset based on their morphological brain views. So, after learning the intact connectomic space, we learned the multi-view morphological similarity SC between subjects across views by maximizing its dependence with the learned intact space X. In other words, we aim to connect the data points based on their locality derived from the learned similarity matrix, so that only the nearest neighbor's observations of a specific point can be connected to this point rather than all other observations. To capture the shared traits across morphological brain views, we use the following formula, where the first term requires that only the nearest sample is assigned as the neighbor of xi with probability 1 and all other data samples with probability 0. Additionally, when the data samples are largely corrupted, the recovered intact space may not be perfect. So we include L1 instead of L2 regularization term in order to handle noisy samples. By solving the first term alone, we may get a similarity matrix equal to an identity matrix. To avoid this problem, a second regularization term for the similarity matrix is added. Since the connectional similarity matrix SC is derived from the intact connectomic space X, we combine both models of intact connectomic space learning and multi-view morphological similarity learning into a joint alternating optimization framework where the learning of the intact space is influenced by the learning of the similarity matrix and vice versa. We note that the stability term which regularizes X is discarded since it will be automatically regularized by similarity learning. So the last step of our framework is ICBM learning. So here we fit a linear mixed effect model described by this equation, where Y denotes the phenotype vector that describes the clinical state of samples, for example, healthy or disordered subjects. Sigma is the covariance matrix that contains data of covariate variables such as age and gender. Fe is the fixed effect vector. Re, a random effect vector resulting from a zero mean multivariate Gaussian distribution with a covariance matrix ST. And epsilon denotes the noise vector with variance VE. This results in using the generated similarity matrix along with phenotype vector and the covariance matrix to compute intact connectional brain morphometricity MC as follows. So this is the formula. Here VA is the variance captured by the estimated similarity matrix ST, and VC is the phenotype variance, so that the proposed ICBM can be described as the proportion of phenotypic variation that can be explained by morphological brain connectivity. In our experiments, we used 155 autistic subjects and 186 normal controls from the ABI dataset. We used 4 cortical measurements in the right and left hemisphere: mean maximum principal curvature, mean cortical thickness, mean circle depth, and mean of average curvature. We generate different combinations of brain network views. For example, using 2 views, we estimated C4-2 combination to generate their ICBM. Then we average all these obtained ICBM across all pairings along with the standard deviation to get the final result. So here we report ICBM estimation when tuning the parameters for the left hemisphere and then fixing them for the right hemisphere and vice versa. So for example, in the left figure, we estimate ICBM for both hemispheres while fixing the model parameters using the left hemisphere. In the right figure, we estimate ICBM for both hemispheres while fixing the model parameters using the right hemisphere. We can see that both figures exhibit similar trends where the estimation of ICBM for the right hemisphere is higher than the left hemisphere for 3 and 4 view-based combinations. As for 2 view-based combination, we note that the estimated ICBM is invariant across cortical hemispheres to some extent. Our preliminary findings show that autism is not significantly morphometric on a connectional level. However, the right hemisphere is more morphometric than the left hemisphere, as we can clearly see that ICBM of all right hemisphere combination of views is higher than the left hemisphere. Also, we notice a huge difference between left hemisphere and right hemisphere ICBMs for the combinations of 3 and 4 views. This might indicate that the complementarity between multiple views, specifically where the number of views is higher than 2, is nicely captured to learn the intact space. In this paper, we presented the first work introducing brain connectional morphometricity. We evaluated the proposed metric on brains diagnosed with autism, and specifically, we learned an intact connection on brain morphometricity using multi-view morphological brain network data. Our ICBM showed that autism is not significantly morphometric on connectional level. We also discovered that the right hemisphere is more morphometric than the left one. However, these preliminary findings need to be further investigated using larger autistic data sets. In our future work, we will evaluate the proposed ICBM learning framework on other disordered data sets, for example, dementia, and compare it with the conventional brain morphometricity. Thank you for your attention.
For more, please visit us at basira-lab.com And don't forget to subscribe, like, share, and cite our work. Thank you.